Hello students, my Shoaib sir. Today I shall discuss one of the most interesting topic of class 12 biology, that is the molecular basis of heredity. You know what is heredity? Student, heredity means the transmission of biological characters from one generation to another generation, then further in future generations. That is called as heredity. These characters are traits inherited by the progeny from their parents because of they inherit some particulate substances from their parents. When the biology is not much advanced, at the time people got puzzled. What is that substance that progeny inherited from the parents? There are two categories of biomolecules actually in the mind of researchers that could contribute for the heredity. One, protein. Second one is nucleic acid. Do you know why people considered protein should be a hereditary characters? The answer is very simple. Because protein is the most diversified biomolecule in the living world. You also know the biodiversity, enormous biodiversity. If you match the biodiversity in the term of volumes, it can only match the diversity of biomolecule, that is protein. That's why people got puzzled and strongly advocate protein as the hereditary molecule. On the other side, nucleic acid is advocated as hereditary substance because nucleic acid is the only substance which can self-replicate. That means one nucleic acid to another nucleic acid. Half of them is conserved and half of them is newly built up. That later on we shall discuss. The semi-conservative replication. But today our topic is how the molecular basis of hereditary material is established. There was a strong debate, as I told you. Somebody said protein, somebody said it's nucleic acid. So people supported them involved in investigations to prove their logics, prove their claims. So several people demonstrated different type of experiments. The most significant experiment came from one is Griffith's experiment. You know Frederick Griffith? Frederick Griffith used the mice as an experimental animal and two batches of pneumonia. One is R strain, another is S strain. So two batches, one is R strain, another is S strain. Virulence means this S strain, when infect an animal, in this case mice, it not only replicate, increase its population size, it also release a toxic substance, and that toxic substances causes this rat pneumonia. If it got acute, then finally the rat dies. In his experiment, all the rats injected with virulent strain or S strain, they developed pneumonia, not only pneumonia but died. On the other side, another strain, R strain, non-virulent strain. This non-virulent strain, they replicate, increase their population, but they do not release any toxic substance. So rat do not develop pneumonia. So you got clear? One R strain, another S strain. S strain causes S strain is responsible for causing pneumonia in case of rat. R strain not. This is very clear. Now, you know what I told you. Whenever S strain infect the animal, it replicates very fast. It increases its number and release the toxic quantity to a lethal extent, lethal dose that causes pneumonia and death. If the animal is dead, suppose we shall heat it and kill them, that is called as attenuated. Simply, you know very well, pasteurization, heat the milk to some degree to check 60 degrees Celsius to check the growth of microorganisms. Similarly, if you heat it, the bacteria will be killed. Once the bacteria will be killed, then if you inject that killed bacteria into the rat, what will happen? Nothing, because it can't replicate. It can't increase its size. It can't release the toxic substance to a lethal dose. So whenever the rat injected with attenuated S strain, Griffith found the rat without any disease. They are jumping here and there is no problem. But one thing very interesting, students, listen. This is harmless, R strain is harmless, attenuated S strain is harmless. Both are harmless. Individually, when Griffith injected either R strain or attenuated R strain, all their batches of rat did not develop any disease. Remember this point. But what is amazing? The amazing fact is, whenever these two harmless, that means it is made harmless, by born not harmless, it is made harmless, and it is by born harmless, when that mixed and injected to the this, what are S strain plus R strain, that is mixed, then injected. Amazingly, what happened? The rat developed pneumonia. Griffith surprised. Then he started investigation. He knew very well both were harmless. Still, that rat developed pneumonia. He investigated. He found live S strains in the blood smear of the diseased, yani pneumonia caused rat. Understood? What Griffith did? He injected killed a strain. As a student of biology, you must know the golden line or the golden principle of biology. anything is dead, it cannot retrieve. It cannot revive. Neither it can replicate, neither it can reproduce, nor it can release the toxic substance in a significant quantity. But very interestingly, what he found? He found live S strains in the blood smear. So, to the logical mind of Griffith, one conclusion he got. What is that? Dead cannot retrieve. Only living can reproduce and release the leaving the toxic substance. That means there is one possibility. This possibility is out there, because living cannot revive. Then what is the possibility is there? There is a possibility that life's R strain. Evolution. What evolution says? Evolution says whenever the organism born, they born with a difference. They can vary, they can modify. So the R strain is modified into S strain. That is the conclusion. But to his mind, whenever he injected the simple R strain, this transformation of R strain into S strain didn't happen. But whenever he mixed these two, the transformation happened. That means only one conclusion comes from this observation. What is that? Some factors that is released by the dead, originated from the dead, originated, not released, because it is dead, so it can't release, originated from attenuated S strain, might have had taken up by R strain, responsible for its transformation. So Griffith concluded some factor had its origin from dead S strain is ultimately responsible for R strain transformation. So he says this as a transforming principle. So this is all what Griffith did. Griffith did not know what is the physical basis of transforming principle. Later on, MacLeod, McCarthy, Avery, they experimentally demonstrated and found it is the nucleic acid. It is the nucleic acid had its origin from S strain is actually responsible for the transformation of R strain into S strain. That means the physical basis of transformation, the principle is nothing but nucleic acid, nothing but DNAs. Have you remembered this Griffith's total observations and experiment and its conclusion? Okay then, just go through the revision and we shall come back.